presentation will be by Gabriel Oliveira about wood anatomy of the lianas used in ayahuasca.
Yes, exactly. So, hello to you all, and thank you for being here. Thank you for participating in this awesome conference and letting me be here talking about some wood anatomy and botany. So, this project is part of a larger project, as Camila have already said. I am going to talk about the emphasis that we put in the anatomy of the lianas used in production of the tea. So, let's get on with it. But firstly, what are lianas? Well, lianas, simply put, are woody vines, meaning that they do have bark, such as trees or any other plant that has bark, but they have this scandent habit of vines, meaning that they start the development into the ground, and as they develop further, they lose the ability to support themselves, meaning that they have to climb and rely on other vertical surfaces in tropical forests being trees. You can see some lianas in this picture that I'm showing you here, but I think these are better. So the first image I put there is to show you how good they are, how efficient they are in climbing trees and forming this complex network of plants in the tree cups. They climb and twine around trees and even go to other trees to help the climbing of them. The second image, as you can see, shows the different shapes and forms that these lianas can have, and that will help them be more efficient in their climbing. The last image is going to show you how they have a bark that's very similar and has a resemblance to the bark of the tree that is climbing. Regarding the anatomy, lianas, as any other plant, have an anatomy that goes in relation to their habit. So they have a scandent habit, which means that they have to be flexible because they need to twine and make all those moves to be efficient at climbing the trees, and other surfaces, of course. They also need to have a very strong stem, I'm sorry. Because they need to support the weight of their own bodies and bodies of other lianas that might climb upon them. These plants are known to grow up to 100 meters, so you can imagine the weight that they have, so they need to be very strong. And also, they need to have a very efficient conductive system, because the conductive system is going to take all the nutrients and the water that these plants need from the roots to the leaves and vice versa. So, about the flexibility, they tend to invest in a higher quantity of axial parenchyma, which is a tissue that is softer. So having a softer tissue in abundance might lead these plants to have more flexibility. In contrast, fibers are not that they are neglected, but they have fewer quantity if compared to regular wood, of course. Fibers are hard, so if they had much more fibers that they already have, they would be able to support themselves a little bit more, but they would become too brittle. So that's not something that you want if you want to twine and make all those moves. To have more strength, hard and soft tissues are intermingled in the stem, and that helps them have more strength. These hard and soft tissues might be the axial parenchyma being the soft and hard being the fibers, or they might have other tissues, for example, the phloem and xylem that I'm going to talk about more specifically later. The wider conduction cells are present in the xylem and the phloem of these plants, and that is that if you have wider conduction cells, then you're going to have a more efficient conductive system, and that's precisely what they do. These characteristics, all of them summed up, form what is known as the liana-scent vascular syndrome. You might want to recall the name later. As expected, these plants have roles that goes in various ranges of areas. So, for example, they might have an ecological role, as you can see there, providing shelter and also food, and also acting as a means of mobilization and transport for various animals, such as mammals, birds, or even insects. But they might also have a role that is more economical and social, being the raw material for handicraft and production of furniture in various communities. As talked about Scipione, there is a community that relies basically on Heteropsis lianas for the production of those materials, as I said before. But they have, and that's the main point here, another important role, that is that they are the raw materials for the production of ayahuasca. So tucunacá and caupuri, most precisely, which are the lianas I'm going to emphasize here. Because of the various uses that these plants might have, they need to have very important and concise taxonomical research done to them. Because if you know, then you can control the quality and the veracity of these plants. Because of the widespread use and sale of these plants, how can you assure that the plants that you're buying are, in fact, the plants that you're seeking? So if you don't know the taxonomy, if you don't know the classification and a way to identify these plants, accidents might happen, leading into death even. Because if you try to produce a tea that's not with the plant that you're looking for, then what if that plant has some toxic chemical? So that might lead to death. And also to an extinction of these plants, and even extinction of cultures. Unfortunately, taxonomical researches are not very common nowadays. Tucunacá and caupuri are mainly the two lianas that are related in scientific community. If you search, then you will find a neglect of other lianas that are known by traditional sources to be used in production of ayahuasca, but all neglected or very little mentioned about them. So, and even tucunacá and caupuri, they are the ones that are mentioned. There isn't a consensus in the scientific medium. Some people say that they are two different species. Some people say that they are the same species. So there is not a consensus, and that might be a problem later. So the objectives of this study and all other studies are to observe the anatomical features of these plants, from an anatomical point of view, of course, caupuri and tucunacá being the two plants that we looked on, to compare the traits between these two lianas, also to study the similarities of these plants, the similarities and differences, of course, and try to complement and to enhance the knowledge in an anatomical point of view. To do so, we gathered 5 samples of these plants, 2 of them being from caupuri and 3 of them being from tucunacá. We identify them by the external morphology. Camila has already said something about their external morphology, but I'm going to talk about it more now. Caupuri has these circular nodes that are very characteristical, so you can use that to identify these plants. And also tucunacá has lobes, cylindrical lobes surrounding the stem. We collected 2 caupuri from 2 different ayahuasca groups in Brasilia, and the tucunacá we collected 3 of them from 2 plantations in 1 gallery forest in São João da Aliança. Both lianas were analyzed macro and microscopically following the Yawa list and also Angelosi. The results were somewhat expected in some areas, meaning that these plants have the lianas intervascular syndrome, as I mentioned before, meaning that they have very wide vessels, as you can see in that image. Those vessels are very characteristic traits of lianas. They also have more axial parenchyma than fibers. I'm going to show you later that. And also cambial variants. Remember when I said the hard and soft tissues might be intermingled? Well, this is the case. The interxillary phloem is the mixture of hard and soft tissues, being xylem the hard tissue that commonly is made by the cambium, which will produce xylem to the inside and phloem to the outside. But here, that doesn't happen this way. They do produce xylem to the inside and phloem to the outside, but there is a variant in the cambium that will produce the phloem to the inside the xylem. That's an important characteristic, and that is shown in the white arrows, as you can see there. There were other features. For example, regarding the vessels have vessel dimorphism that is very more notable closer to the bark, as you can see there. And vessel dimorphism, as if you take a look at the red and the red circles, as you can see there is larger and there are smaller vessels side to side, and that's a very important characteristic to mention. Regarding the axial parenchyma, we found 3 types of axial parenchyma, meaning being them the scanty axial parenchyma, which is the black circle. Scanty means that there is very little axial parenchyma surrounding the vessels. There is vesicentric, which is shown by the arrows. And vesicentric is the axial parenchyma that is completely surrounding the vessels. And there is also irregular lines being formed by non-lignified axial parenchyma. Those irregular lines are also something interesting to look at because they form only from a certain distance from the pith. And from that distance on, they start to grow larger and larger, forming these marginal bands, as you can see there. Prismatic crystals were also identified. The prismatic crystals are those thingies that are on the axial parenchyma, those prisms, and they do appear in the 2 lianas that we found, that we observed. And there were also vestured pits in the vessels. Pits are the way that vessels communicate and pass nutrients and water from one vessel to the other. And being vestured, that is some sort of reinforcement in the pits is a very characteristical and notable feature as well. All of these characteristics that I'm shown here are similarities, but they do have differences, and those differences are most precisely the frequency and the size of the vessels, being Kaupuri having frequent narrower vessels, and Tukunaka having wider vessels, but with less abundancy. These 2 pictures are on the scale, so you can compare the size and the frequency of these vessels. Regarding crystals, Kaupuri has a large frequency of drusas, which are crystals that look like stars, and they do happen in the interxillary phloem. They are existent in the Tukunaka as well, but they are in a very smaller quantity, and they also come in small sizes. Crystals cluster radially in the axial parenchyma in that radial organization is something that only appears in Tukunaka, so that's another thing to look at. And lastly, the last difference is regarding the parenchymatic series. That series is how many cells there are on top of one another, and inside that object that I put there in red, you can count the cells and see that Kaupuri has 2 to 4 cells per axial parenchymatic strand, and Tukunaka has up to 8 cells. That was the maximum that we could count. So, what can we say about those differences and similarities? Well, these 2 lianas are indeed related because they share qualitative features, but they have some quantitative features that are not conclusive. You can say that they are 2 different species, but you can doubt because they have different quantitative data, they have cambio variants, they are different, they form different patterns, and that is not saying that these 2 lianas are different, but they might be differentiated in the future. That's why there is further research being done, not only by an anatomical point of view, but as Camila said, there are 5 labs working in this, so there will be more in the future, there will be more conclusive conclusions. So I want to say some thanks to these people. They made this project possible, and they were advising us each step of the way. And also I want to thank each and every one of you for being here, paying attention, and forming this great conference. Thank you all.